Hi, my name is Sudithi, and my biology role model project is on Dr. Jennifer Doudna. Dr. Doudna grew up in Hawaii, and although she had no direct family members who were scientists, she was encouraged to pursue her passion after discovering her interest in the field during a high school chemistry class. She also read The Double Helix by James Watson, which interested her in the subjects of DNA and RNA. She pursued this goal by getting a bachelor's in chemistry at Pomona College, and then a master's and PhD in biological chemistry and molecular pharmacology at Harvard University Medical School. She then continued to work at University of Colorado Boulder and Yale University, after which she moved to UC Berkeley, where she still actively does research while teaching as a professor. In one of her interviews after winning the Nobel Prize, Dr. Doudna says a main challenge she faced was overcoming the criticisms women in science face, and she encourages girls in STEM to pursue their passions no matter the backlash. Some of Dr. Doudna's hobbies include attempting to grow tropical plants from her home state of Hawaii in Northern California, which she regards to be a puzzle. She also enjoys reading. I admire and relate to Dr. Doudna as I have always been interested in research regarding the life sciences. I have also grown up in Northern California, close to UC Berkeley, so I was introduced to her work briefly many times in biology classes, making me more interested in her work now. As I explained when discussing her background, Dr. Doudna specifically focuses on RNA, and has, in fact, pioneered the research that led to the development of CRISPR-Cas9, a technique used in genome editing. This is one example of a recent scientific paper Dr. Doudna is an author on. It is titled Structure-Guided Discovery of Ancestral CRISPR-Cas13 Ribonucleases. This paper explores the systems of the ribonuclease Cas13 and its ancestor Cas13N in order to expand the ability to edit RNA. Cas13N has a singular active site where RNA-guided cleavage occurs, as well as CRISPR RNA gets processed. This means that studying it allows for the understanding of its evolution. The image, titled Figure 2 in the paper, shown on the slide, shows how Cas13N has evolved by using E. coli, the bacteria, as a host with the Cas13N plasmid. After running a procedure to sequence RNA, researchers were able to determine that this was an ancestral trait of current Cas13, as seen in Figure 2a, as only a specific sequence was able to be found. This short sequence is specific to RNA. Additionally, Figure 2b shows that green fluorescent protein, or GFP, was used to determine the specificity of Cas13N, which again relates to Figure 2c. This part of the figure shows how a serial dilution was used to observe how the GFP was present in cells and dimmed with Cas13N due to the specificity. However, RFP did not seem to decrease as it was not targeted by this ribonuclease. Figure 2D and 2E are also related as they investigate Cas13N's ability to prevent against infection by phage. Figure 2E reflects that Cas13N could not attack DNA, only RNA, which is further reflected through the chart done in Figure 2F. Figure 2F shows how different viruses' RNAs were attacked by at least one cRNA, but none of their DNAs were attacked. Dr. Doudna's work has led to breakthroughs regarding genome editing, and one of her next steps is to expand this directly to the human body while exploring the effects on human genetic variation on Cas and CRISPR-19. This can be helpful as it leads to many medical breakthroughs as well as helping in fields such as agriculture. Thank you for listening to my presentation, and here are some of the citations that I used throughout my presentation.